So we do not see very many diseases of brassicas in Utah. Either we don't have them or you guys haven't sent any samples yet. So I'm going to talk initially about the few diseases that we do see, and then I'm going to talk about some diseases that you might see based on either the pathogens are in the state on other crops, or they could be brought in on seed, or I've seen them on weedy brassica species. So what have we seen in Utah so far? We do see damping off and root rot. As in many other vegetables, seedlings or young transplants, they just turn brown and black at the base and then they literally fall over. Causal agents are usually different species of Pythium or Rhizoctonia solani. This can both be a problem in the greenhouse, in transplant production, as well as in the field. So down here on the left, you see some seedlings that are decaying and dying in a seedling tray. If you have a tray of seedlings that looks like this, you have individual plants dying, you most likely have to throw all the plants away, because those that still look healthy are most likely infected and they just don't know it yet. Here in the middle, this was a trial I had a couple of years ago with cauliflower. This is a drone image that we took, and you can see a couple of plants that died. And when we isolated the pathogens from the root rot that we saw, these plants were about 4 weeks old. We isolated Pythium. So this is one way where a drone comes in really handy, especially if you have a larger area that you want to survey. It's very easy to see Pythium in the field. And then on the right here, this was a sample sent in from a grower. The radishes had these black lesions, and this turned out to be Pythium as well. And that was in a early summer when we had a lot of rain and their field was very wet. So they lost a lot of their radishes to Pythium at that time. Usually you can find Pythium and Rhizoctonia in fields that already have a history of the pathogens. These pathogens are very indiscriminate. They take any vegetable seedling they can find. You could mostly find them during very wet spring and early summer, because both pathogens like it wet, especially Pythium. Pythium has spores that can swim in a film of water. They have flagella, which are hair-like appendages that they use like a propeller to move themselves forward in a film of water. In a greenhouse, as I mentioned, you can have contaminated trays or pots that you reuse. The potting mix could be contaminated if you mix your own potting mix, let's say on a floor without any plastic. Benches can be contaminated with the pathogens. And if you use water in your trays that you keep recycling, like a hydroponic system, Pythium can survive in that water, and you could recycle the water through all your trays, spreading the Pythium. To manage damping off and root rot, sanitation is your best friend. So disinfect trays, pots, and benches with a 10 to 15% household bleach solution. Pots and trays should be soaked for at least half an hour to an hour in the bleach solution. Afterwards, you want to rinse it very well with water. You don't want to have any chlorine residue, because that could cause phytotoxicity. If you do use a holding tank for your nutrient solution in hydroponics, you want to regularly clean that tank. Also, do not overwater seedlings in those trays. If the seedlings are sitting in water, they're more susceptible. Do not plant the seed too deep. That just has the plant being exposed to the pathogen in the soil longer before it breaks through the surface. Seed treatments with fungicides can prevent damping off. So if you can either do the seed treatment yourself or something like Thyram, or you can buy already treated seed, that would be your best option. Now, powdery mildew, we've seen that occasionally all brassica species are susceptible to it, including radishes and also weeds. And I've seen it quite frequently on wild mustard. I've seen it on radishes. The species is Erysife cruciferarum as well as Erysife polygoni. They're fairly host specific to brassica species. But if you see wild mustard that has powdery mildew and you grow other brassicas, you need to be watching your plants very carefully to see if anything starts on your crops. Now, powdery mildew, in contrast to many other fungi, does not need free water on the leaves to infect the plants. And rain can actually have a negative effect on the fungus, as does overhead irrigation. However, if it's very wet and you'd use overhead irrigation, you might invite other fungi to come in. The spread of powdery mildew, there's several different ways. The spores are carried by the wind for miles. Infected plants that contact non-infected plants. So if you have an older crop and then next to it is a younger crop, if those leaves touch, the spores fall off very easily. And then they can cause new infections on the new leaves. And also dispersal by humans. If you walk through your plants and they have powdery mildew, the spores will be attached to your pans, and then you walk and brush by your other plants, and then you deposit the spores that can then cause infections. To manage powdery mildew, use resistant varieties if they are available. Remove infected plant material at the end of the growing season to prevent the overwintering of the fungus. Control weeds such as wild radish or pepperweed, since they could also carry the spores. We frequently see it on wild radish. They can also have the survival fruiting structures throughout the winter. And then next spring, if you plant new brassica, the spores will be right there to infect your plants. There are some chemical control options as well, such as strobilurins like Caprio or Quatros, for example. You can use sulfur products, but do not apply them above 90 degrees Fahrenheit. You will fry your plants. So either apply them very early in the morning so that there's at least 2-3 hours before you reach 90 degrees Fahrenheit, or late in the evening after it cooled down below 90 degrees Fahrenheit. And of course, you always have to follow the label for concentrations. Application should start when the first powdery mildew spots are detected on the leaf. So here on the bottom left, you see like individual colony on this tomato leaf. That's when you want to start spraying. If you wait until the leaves are completely covered, like here on the right, you will not get any control anymore. And you need to apply the fungicides about every 7 to 10 days. That's how long it takes from the initial infection, that's the spore germinates, colonizes the leaves, and then the new lesion starts producing spores. So you have to keep up with that reproduction cycle all summer long. Verticillium wilt is another pathogen that we see. It has a wide host range. It likes a lot of vegetables, including tomatoes, peppers, eggplants, as well as brassicas. It also goes to alfalfa. So if anybody rotates with alfalfa, there is a possibility that you have verticillium in the soil as well. In the brassicas, it affects cauliflower, cabbage, Brussels sprouts, horseradish, rutabaga, and radishes. It's spread by spores that are produced on the plants, but also microsclerotia that survive in the soil. They can survive for up to 20 years in the soil. And you can see some microsclerotia here in this picture on the bottom left. They're small black hard balls of mycelium. And the black color, it's melanin that prevents the sclerotia from being attacked by other soil microorganisms and being destroyed. So this fungus can sit in the soil for years waiting for a suitable host plant to come along. Here are some pictures of symptoms. Here you can see the initial yellowing of leaves in a cabbage field. And then also if the disease progresses and you cut the stem at the base, you can see the vascular discoloration caused by Verticillium. Management of Verticillium is difficult. You want to make sure that you clean your equipment to prevent moving Verticillium from an infected field to a non-infected field. So you want to wash off the soil, for example, or any plant debris where sclerotia or spores could be hiding. One brassica species that's actually immune to Verticillium wilt is broccoli. And in some studies, they have shown that incorporating broccoli residue in the soil may reduce disease severity over time. They incorporated it at a rate of about 2.5 tons per acre. So in that case, that study was done in California. They were able to get the leftover brassica residue from a processing plant, and they incorporated that. An interesting virus that we came across in Utah was turnip mosaic virus. It was on horseradish down in Caseville, and it has a wide host range. It goes to many brassica species. It goes to ornamentals, beets, lettuce, spinach, and also some weeds. But so far, we've only seen it on horseradish. It did not affect any of the other brassica species that were growing near them. And what you see is you get these yellow spots with a green center, or you get a mosaic pattern on the leaves. It's transmitted by aphids in a non-persistent manner, which means the aphids feed on an infected plant. The virus particles are stuck to their stylet, and then they move to a healthy plant. They feed on that healthy plant, and in the process of feeding, they deposit the virus particles that are on their stylet. It only takes a few seconds, at the most half a minute, to transmit the virus from one plant to the next. It's therefore important to have good weed control, especially since a lot of weeds do not show symptoms. Weeds learn to tolerate a lot of viruses. They can grow just as well with virus infections, in contrast to our crop plants that oftentimes do not get along with the viruses. You also do not want to plant susceptible crops next to perennial brassica species like horseradish, since that horseradish can be the overwintering source of the virus. Now I'm going to move on to some diseases that you may see in your Utah brassica crops. One is black rot caused by the bacterium Xanthomonas campestris. The symptoms initially that you see are yellow V-shaped lesions resulting in necrosis, and you can see that here on the left. And then you also later on see blackening of veins. It's transmitted by seed, splashing water, equipment, and insects. Disease development usually occurs during warm and humid weather. Normally, Utah, the weather is very dry in the summer, but if you have overhead irrigation, that equals humid weather in many cases for plant diseases. The bacteria can enter through wounds or hydathodes. Hydathodes are pores in the leaves that the bacteria can enter through. This is why you often see it in that those V-shaped lesions. That's where hydathodes are in the area, and that's where the infection occurred. The bacteria then move through to the stem and then start colonizing the entire plant. And seedling infections may go unnoticed initially because of temperatures that are too cool for symptoms to develop. Management, sanitation, you want to remove any plant debris or diseased plants where those bacteria could survive. You want to avoid overhead irrigation, as splashing water can spread the bacteria. Purchase disease-free seed. If you can buy seed where the company says that they tested for certain diseases, and usually they list the diseases they test for, the seed might be more expensive initially, but it may save you a big headache in the long term. Not only for that one crop that might be infected, but it's also a way to introduce diseases permanently into your operation. Use resistant varieties when they're available. And also some farms have used hot water treatment. You have to be careful with hot water treatment because some varieties are more sensitive to hot water treatment than other varieties of cabbage, broccoli, or other brassicas. So you might want to try it first and see if you lose any germination ability of the seed. So you would use 122 degrees Fahrenheit for 25 minutes for cabbage, broccoli, and Brussels sprout seeds. And then 15 minutes at 122 degrees for cauliflower, kohlrabi, kale, turnip, and rutabagas. And that should destroy the bacteria. Bacterial soft rot, we see that here on other crops. It's Erwinia carotovera. It's another one of those pathogens that takes any vegetable that is soft and juicy or easily accessible. Usually if the fruit is lying on the surface, comes in contact with the soil, you can see Erwinia carotovera as well. It's transmitted by splashing water, insects, and equipment. The disease develops again during warm and humid weather. The bacteria enter through wounds that are either created by insect feeding or by damage through equipment use. The bacteria can survive in plant debris. And it's not only a field problem, but symptoms can also develop in storage. So if you harvest your cauliflower or broccoli and the knife you use to cut the heads have the bacteria on them, you can introduce the bacteria to that cauliflower or the broccoli head, and then it will start rotting in storage. The symptoms you see early in the season are usually water-soaked lesions, but then the disease quickly progresses and you see a mass of macerated tissue with cabbage internally. So the outside of the cabbage head looks still intact. It might be a little bit flattened on the top, but then if you cut it open, it's all rotten on the inside. And occasionally you see slime oozing from the cracks. Erwinia carotovera produces enzymes called cellulases. They dissolve cell walls. So you get all the plant cells to disintegrate, and the contents of those plant cells pool in the center, and that's the ooze that is coming out of the heads. You also get hollow stalks in cauliflower and broccoli, and the heads of broccoli and cauliflower have brown sunken areas. So here you can see an initial infection. You see this brown necrotic lesion on the outside of a cabbage head. Eventually this disease can progress and you see the water soaking, and if you would cut that head open here, that Napa cabbage, it would probably be fairly liquid on the inside. Here's another cabbage head that has advanced symptoms. And then up here, this is what the cauliflower head would look like if it's infected with Erwinia carotovera. Management of Erwinia carotovera is sanitation. You want to decontaminate knives that you use for harvesting quite frequently. If you just wipe them with a Lysol or Clorox or other disinfecting wipe, or if you have an alcohol solution that you could use to disinfect the knives, especially if you had problems with Erwinia carotovera, either on other brassicas or other vegetables in the past. Remove any cull piles of vegetables that are nearby, because that's where the bacteria would survive, and then rain or insects could move the bacteria to your crops. Rotating with small grains for a few years can help. Small grains are not susceptible, and if you can use those small grains until the old crop residue from your brassicas has decomposed, and then the bacteria will not survive or not be able to survive very well in the soil. Use well-drained soils or raised beds if you had problems with the disease in the past. There are some tolerant varieties of broccoli that are available, and it's broccolis that have a more cone-shaped head, because there's less chance of water pooling on the heads where the bacteria could multiply and then cause an infection. I'll talk about blackleg. Blackleg is a fungal disease caused by foma. It's another pathogen that could be seen. It's a seed-borne pathogen. Sometimes you can see lesions on the cotyledons. They will develop small spots on the leaves. And in severe cases, the seedlings will die, and it looks just like damping off you get from Pythium or Rhizoctonia. Older plants, the lesions have developed very defined, they're confined by the veins, so they will never cross the veins on a leaf. So we'll see individual lesions, and then you can see the fruiting structures of Pycnidia that develop in those lesions. They're little black spots. They look like pinheads. So here you can see an initial lesion, and you can see here's the vein of the leaf, and it's confined to this area between the veins. And on this picture, the spores are there, but you would have to zoom in to see the fruiting structures here in the center. Here's a close-up of the fruiting structures, these black small spots. You can see them with a strong hand lens. And then eventually the fungus will colonize the vascular system and then starts rotting the roots, and you can see the root decay. The fungus does survive in crop debris, and as I mentioned, is seed-borne. Spores germinate on the leaves, and then they colonize the plants through the petioles, and then move from the petioles towards the vascular tissue where water and nutrients are taken up, and then they transport the fungus throughout the plant. Spores are released at about 46 degrees Fahrenheit. An infection occurs and the symptoms develop rapidly after 68 degrees Fahrenheit. You will not see any symptoms even though the plants are infected if the temperature is below 50 degrees. Management, again, use disease-free seed. Hot water treatment can help in this case as well, but you want to follow the same rules that I mentioned for black rot before. Remove crop residue to remove the overwintering fungus, and crop rotation from 3 to 4 years to non-host plants can also be useful. There are some chemical control options. Again, you can use strobilurins like Caprio following the label rate. And white rust, I see that quite frequently on wild mustard. It's actually an oomycete. Even though it's called a rust, it has nothing to do with the rust fungus. It's different. It looks similar, but it is an entirely different pathogen. You see white pustules develop on above ground plant parts. Pustules may merge, and then you have a larger area of blisters. Leaves might curl, and flower heads get distorted. So here are some pictures. You see these pustules on leaves. Above the upper side of the leaf, you will only see chlorotic spots. You then have to turn the leaf around and then you see those pustules. Here's those pustules on some radishes, on some cabbage. And then when the flowers get infected, you get these distorted flower development, and for seed crops, you will not get any seed production. And since these seed or flower stalks often look similar to antlers, they're also called stack heads. Transmission is through rain or splashing water, and it can also be seed-borne. The disease survives as oospores on seed and in soil. The oospores release motile zoospores. They can swim in a film of water, just like Pythium. They're related to this group of pathogens. And they cause initial infections of seedlings. The new infections will then form a different type of fruiting structure, sporangia, that is in those white pustules, and they are then wind dispersed and cause secondary infections within your crop. Temperature range for infection is between 55 and 64 degrees. To manage the disease by disease-free seed, crop rotation, this pathogen is specific to brassicas. Good weed and volunteer brassica control, like I said, we see it quite frequently on wild mustard. And you can use the chemical controls such as strobilurins, as well as mefenoxam, following the label rate. And that's all that I had.
People have asked, Sarah asks, I had powdery mildew on beans and squash last year. Should I treat the area before I plant this year? She's going to trial some new crops in that area and wants to know if that's going to be a problem.
I would have to look up the exact powdery mildew that goes to beans. If you plant beans again, yes, or any legume, I would say go ahead and clean up the area. If you plant brassicas, the bean powder and mildew won't go to it.
Okay, good, good. There's another one from Steve. He asks, is seed treatment like weak peroxide, hydrogen peroxide, an effective and what he calls a poor man's alternative to fungicides? People ask me that question a lot, and I'd like to hear what you think we should do. Should they use peroxide?
I know there are some products that are based on hydrogen peroxide. They can sometimes cause phytotoxicity, especially if you apply them during hot temperatures. I have not used it, so I can't attest to how well they work. But I would be more concerned about phytotoxicity, that you will burn the leaves.